Hey everybody, welcome to supplementary video covering chapter 2.5. And this one here, we're going to be talking about organic compounds. A lot of it probably will sound familiar from biology. It's basically, we're going to be going over the 4 macromolecules in here, but this is the final video lecture for Unit 1. So organic compounds, they're going to have hydrocarbons, so that any extra bonding space that a carbon might have after it's attached to all the functional groups is going to use hydrogens to fill those points. Remember, each carbon atom can form 4 covalent bonds. So functional groups that a carbon might attach to are these down here below. These functional groups, they are going to be commonly found attached to a carbon, and they like to behave as if they were all one thing, or they like to behave as if they were their own molecule. And when things like to break on an organic compound, these functional groups like to stay together. So you see hydroxyl, or an OH group is a common one in your alcohols. It's a polar molecule, and it's very important. Remember from the last video, it's important for dehydration synthesis and hydrolysis. Your carboxyl groups or your carboxylic acid groups, they're going to have some sort of molecule attached to a carbon here, and then this carbon is going to be double bonded to an oxygen and then attached to an OH group on this end here. So we see this a lot in different types of acids. This is a carboxylic acid, carbonyl group here. We're going to see them commonly in amino acids and fatty acids. Amino groups are going to be anything with an NH2. Usually you're going to have your carbon over here attached to the nitrogen on this end here. You're going to find those in amino acids. Methyl groups are just a CH3, the most basic, one of the most basic groups you can have. It's going to be found in amino acids. And then your phosphate groups are PO4 2-, which we're going to see in phospholipids and nucleotides. So carbohydrates, one of the 4 macromolecules. The most basic form of a carbohydrate is a monosaccharide. You can put 2 monosaccharides together to get a disaccharide, or you can put as many monosaccharides together as you want to form a polysaccharide. So your monosaccharides are going to be the hexose sugars, which are glucose, fructose, and galactose, as well as the pentose sugars, ribose and deoxyribose. So hexose sugars basically means they have 6 carbons, or they have a hexagon shape. You'll see that on the next slide here. Pentose sugars, those are going to have a 5-sided shape. They'll have carbons and usually an oxygen involved in them, if I remember correctly, on ribose. Yep. Disaccharides, you're going to get those through dehydration synthesis of your 2 monomers, your 2 monosaccharides. Those are going to be things like sucrose, lactose, and maltose. And then your polysaccharides, these are long chains. Those are going to be things like starch, glycogen, and cellulose. And so all bodies are going to use glucose here, a hexose sugar, to form ATP in the process of cell respiration. We talked about that extensively in biology here, where you're going to have a ribose sugar, and adenine nucleotide, and 3 phosphates in this ATP, but it's all coming from the chemical energy stored in this glucose molecule. So here you can see the 3 hexose sugars, your glucose, fructose, and galactose, and then your pentose sugars, deoxyribose and ribose. Over here you can see your disaccharides, sucrose, which is formed from the bond between a glucose and fructose, your lactose, which is formed from a galactose and glucose, and then your maltose formed from, you have here 2 glucose molecules. So glucose, your most pure form of sugar. Fructose we typically see in fruits, and then lactose is your milk sugar, maltose is your malt sugar, sucrose is your table sugar. So the one that you normally see is sucrose. And then down here you have your starch, glucose, and cellulose. Starch has a very basic branch chain here, the amylose and amylopectin. Glycogen has much more branching going on. Cellulose is very tight packing, and which makes it impossible for us to be able to digest because we don't have the enzymes necessary to digest fiber like this. Lipids, they are hydrophobic, meaning they repel water, which can be very useful for creating waterproofing layers. You can have 4 major categories of lipids. You can have your triglycerides. They're definitely going to be the most abundant. You get them from the dehydration synthesis of a glycerol molecule and 3 fatty acids. We'll see what those look like here in a little bit here. But if a triglyceride has no double bonds, like this one here at the top here, it is called saturated. Yes, there's a double bond here from the carboxylic acid, but we're talking in this glycerol, or in this carbon chain here, hydrocarbon chain. There's no double bond, and so this is going to be able to pack very tightly, and so it's going to be solid at room temperature, and we call it a saturated fatty acid. If a double bond exists, that double bond, just based on the chemical shape of that carbon, the bonding shape, that's going to mean that cannot pack very tightly, and as a result, it's going to be liquid at room temperature, and we call it unsaturated. So lipids are your triglycerides are important for fuel sources for your long, slow physical activities, thinking like walking for miles and miles. They're also important for insulation and cushion. Your other categories, phospholipids, they're going to be made of a glycerol, only 2 fatty acids instead of 3 fatty acids, and a phosphate. And those are going to be special because that phosphate is polar and wants to be close to water. It's hydrophilic, but it's being pulled by the hydrophobic tail, that fatty acid tail, and so you kind of get this dual working of that phospholipid, where part of it wants to be is attracted to water and the other part is repelled by water. Another category, your steroids. They have 4 hydrocarbon rings. The most important one is cholesterol. Lots of hormones are going to use cholesterol. And then you have prostaglandins, which are derived from unsaturated fatty acids, which do a number of functions in the body. So here you can see the glycerol attached to 3 fatty acid chains. So this is your triglyceride that you get from the dehydration synthesis to be able to combine those two. Your phospholipids, here's your polar head with phosphorus, your phosphate group right there, PO42 minus. Your glycerol backbone and 2 fatty acid chains, your steroids here, the 4 carbon chains, and then your prostaglandins. Okay, your proteins, they're going to be composed of amino acids. They'll be linked together by peptide bonds, and you get that linkage through dehydration synthesis. There's 20 amino acids, and you can put them in any sort of order you want. There's literally infinite combinations because proteins can be 2 amino acids long all the way up through as long as you want. 11 of those amino acids can be made by the body. 9 of them, however, we cannot make by the body, and we have to get them in our diet. So this is why meat is very important, or legumes and things like that, so that we can get all of those amino acids. All amino acids are going to have a central carbon attached to a hydrogen, and then it's going to have an amine group on one side, that NH2, a carboxylic acid on the other side, and then what makes each amino acid different from one another between those 20 amino acids is what's bonded to that carbon in that 4th spot. We call it the R side chain, R just kind of standing for whatever else might be bonded to it. Each amino acid is going to have its own different R group. Structure of proteins is very important. We cover this a little bit in biology. You have the primary structure of the protein, which is basically what is the order of the amino acids. So is it a valine, a leucine, a methionine? What is the order? And then the hydrogen bonds between each of those amino acids is going to determine the secondary structure of that protein. So is that secondary structure going to be an alpha helix, to where it's a coil, or is it going to be a beta pleated sheet? Beta pleated sheets are more rare in the body. Usually we're going to see an alpha helix in the body as a secondary structure. But that's going to be hydrogen bonds between different amino acids, and then the interactions between your alpha helices with other alpha helices or with beta pleated sheets are, as well as some other bonds, things like disulfide bonds. So with sulfur here, that's going to cause the protein to start folding and twisting on itself to give it its tertiary structure. And then if you take 2 proteins that are folded and twisted on itself and put those 2 proteins together, those 2 subunits together, you get what's known as the quaternary structure. And so proteins have very specific shape, very important to their function, but extreme heat, certain chemicals can cause the protein to unravel and actually make it useless. So this is why you need to keep your body at the right temperature, is so that those proteins do not start to unravel and you start to lose function. So here you can see the primary structure, basically what is the order of your amino acids. The hydrogen bonds between them is going to cause them to form into an alpha helix or a beta pleated sheet. The interactions between those two and the disulfide bonds are going to give it its tertiary shape, and then if you take several of those together, you get its quaternary structure here. So this is an example of hemoglobin, which is a tetra unit protein, where we take 4 of these structures, put them together into the quaternary structure to give us that protein. So protein shape is really important, and another way to visualize just how important that shape is to think about enzymes, which are going to be made of proteins. So you take a reactant or your substrate, it's going to bind to a protein in an active site, so that protein is going to have a specific shape that only allows that substrate into that active site. Because otherwise, think of it like a lock and a key. In fact, one of the classes of proteins is called lock and key fit, where only a certain lock or a certain substrate is going to fit into that keyhole or into that active site, and nothing else will fit in there, or nothing else that gets in there is going to cause any sort of reaction. So that substrate comes into the active site, the reaction takes place, the product is released. Sometimes the enzyme might take that key in it, and maybe that protein needs 2 things. Maybe it's doing a dehydration synthesis and building molecules, and so it needs 2 parts to be able to put together. Sometimes it'll have an active site fit for one of those parts, and once it's bound to that one part, it will change its shape a little bit to make sure that it can fit the other part and pull that in, and then it's able to carry out that reaction. So here you can see an enzyme. Again, it's made of proteins. It has very specific structure. It can pull 2 different substrates in. Maybe it's pulling one and splitting them apart. Just kind of depends. But it has these active sites that are built specifically for those substrates, and nothing else is going to fit. And then the last of the 4 nucleotides, or the 4 macromolecules, are your nucleotides. They're going to have a phosphate group, so PO42-, a pentose sugar, and a nitrogen base. They're going to be formed by dehydration synthesis, as well as some hydrogen bonds. There are 5 different nucleotides that you can have: adenine, thymine, guanine, cytosine, and uracil, and you can see the abbreviations for each. Adenine and guanine are fit in a class known as the purines, meaning they have a double ring structure. Thymine, cytosine, and uracil have a single ring structure, and they fall under the category of pyrimidines. And remember, when you're binding, you always have one purine with one pyrimidine. So adenine always bonds with thymine, guanine always bonds with cytosine. If you're dealing with RNA, you're not going to have you're going to have uracil substituting in for thymine. Nucleotides are used in DNA, RNA, and ATP. Remember, this A is adenosine, so it has that adenine nitrogenous base there. DNA does not have uracil. It's used to store genetic information. You should recall that from biology. RNA uses ribose, does not have thymine. Uracil replaces it. It's going to be used to carry instructions from the DNA. So your DNA is super important. You need to protect it. We can use RNA as a messenger in between where we're trying to go. And then ATP uses ribose, adenine, and 3 phosphates. That 3rd phosphate has lots and lots of energy. It's not very chemically stable, actually, and so that's why it has lots of energy. And so when you break off that 3rd phosphate, you release energy, and the body can capture that, use that however it is, and then it becomes ADP, adenosine diphosphate, because it only has 2 phosphates now. But you can add another phosphate to it through a process known as phosphorylation. Okay, so here you can see deoxyribose and ribose. The difference between those two is merely basically just an oxygen molecule here. You have hydrogen here on deoxyribose and OH group here on ribose. You can see the nitrogenous bases, how they fit, your overall double helix structure of DNA, and then what an adenosine molecule will look like here. You have your adenine bonded to a ribose, your pentose sugar, and your 3 phosphates. When there's 3 phosphates, it's triphosphate, so adenosine triphosphate or ATP. If you break one off, you get less energy molecule. You get adenosine diphosphate, 2 phosphates. You break that one off, you get adenosine monophosphate or AMP, which has even less energy to it. So basically, the further out you get, the more energy is associated with that molecule. So that is our review on organic compounds. Again, that is video 2.5. Most of it probably was a review of biology. Hopefully it was at least. But if you have any questions, make sure you're